We're going to talk about enzymes. So enzymes are biological catalysts, and most of the enzymes are made out of globular proteins. So if you remember when we studied proteins, we looked at two types. You had globular, where it's really folded, almost like spherical, and then you had like fibrous proteins that are just like long. These are the folded ones, so they're all folded up nice and tight. Catalysts, the way they work, what do they do? They are going to increase the reaction rate. They're going to increase the speed at which the reaction takes place, and they do that by lowering the activation energy or providing a new pathway that has lower activation energy. And activation energy is just the amount of energy it takes to get the reaction started. Enzymes are a little bit different than regular chemical catalysts that you would use like in a lab. These are going to be used in the body, so some of those conditions, the reaction conditions, are going to be different. All catalysts, whether they're enzymes or chemical catalysts, increase the rate of the reactions. Enzymes do that a little bit more than regular catalysts will do. So they're going to increase the reaction rates by 10 to the 6. That's huge, or 10 to the 12 times faster than an uncatalyzed reaction. Since they're taking place in the body, they're going to have milder reaction conditions. So chemical catalysts usually require like extreme temperatures. They're going to have high temperature, really high pressure. They can have extreme pHs, either really high or really low pHs. Enzymes are going to break down in your body if that's what's happening. I mean, you're dead if that happens. We have to maintain a fairly constant temperature and pH, otherwise all of our proteins are going to break down. So they need milder reaction conditions. So, temperature below 100, we had atmospheric pressure, and then nearly neutral, like our physiological pH, was just slightly basic, but mostly neutral. Enzymes also have really greater reaction specificity. So they're really, really specific. So for substrates or the reactants, and then they rarely have side products. So they're really made for a specific type of reactant. Capacity for regulation. Right, so catalytic activity is going to vary in response to not just the substrate, but some other conditions as well, some other concentrations of other substances, surrounding substances. And then we'll look at a few different mechanisms for regulatory process. And we saw some of these last semesters too. Allosteric control, covalent modification of enzymes. So how can I change this enzyme a little bit in order to make this work? And variation of the amounts of enzymes synthesized. We talked a little bit about that as well. Enzyme nomenclature. So how do we name enzymes? We are not going to name too many enzymes, but we should be able to look at the enzyme and how it's named and figure out either what it's working on, like what is the substrate, or what does it do? What's the action? So something like aldehyde reductase. Well, that name, a lot of these enzymes are going to end in ase. The ase means you have an enzyme. Aldehyde reductase means you are reducing an aldehyde. So here's an aldehyde. Here's a sugar that has an aldehyde. And when it's catalyzed with aldehyde reductase, it goes from an aldehyde to an alcohol. So that's a reduction reaction. So it's reducing an aldehyde. Urease is another enzyme that we'll see. It catalyzes the hydrolysis of urea. So this one's talking about what is it working on? What reactant does it work on? And not really, talking about how it works. So those are the two things. It's either going to tell you about what type of reaction it catalyzes or what kind of substrate it works on. Alcohol dehydrogenase. This one kind of does both. It tells you you're working on an alcohol, and it's a dehydrogenase, so it's dehydrogenating, which is really just an oxidation. So it's going to catalyze the oxidation of alcohols into aldehydes. So alcohol, that's an aldehyde, or sorry, an alcohol. You're starting with. Let's draw that out for a second. So we have an alcohol. It's going to have like a COH group. See, maybe we have just CH2, CH2, maybe one of these. And then, so that's an alcohol. And then if we wanted to oxidize this to an aldehyde, we'd have a CH3, CH2, C, double bond OH. There's an aldehyde. So you guys are really, really familiar with your organic families now, right? Alcohol to an aldehyde. So these are important to know. These are 6 enzyme classifications that based on catalyzed reactions. So what types of reactions do they catalyze? So oxidoreductase, this is going to catalyze an oxidation reduction reaction, which is really just an electron transfer. So if something is gaining electrons, it's being reduced. If something's losing electrons, it's being oxidized. So an oxidation reduction reaction is catalyzed by an oxidoreductase. Transferase, this is pretty generic. That's going to transfer. That's talking about transferring any kind of functional group will be a transferase, and then you can usually put something in front of that, and that will tell you a little bit about what is it transferring, what type of functional group is it transferring. Hydrolase, that's just for a hydrolysis reaction. We have studied a lot of hydrolysis reactions. The enzyme that would catalyze those are different types of hydrolases. Lyase is when you catalyze a reaction where you're uh, adding or removing groups to or from double bonds. Isomerase, so looking at different isomers. So isomers, if you remember what isomers are, we looked at a lot of those when we were looking at carbohydrates, and we had those enantiomers and diastereomers, those sorts of things, where they have the same connectivity, right? All the atoms were connected in the same way, but they had a slightly different orientation in space. So that's one type of isomer that you can have, and you would break those bonds, rotate it, put it back together, and then that would be the enzyme that catalyzed that process would be called an isomerase. And then we have ligases. Ligases are usually formation, the catalyze, formation of carbon-carbon, carbon-sulfur, carbon-oxygen, carbon-nitrogen by a condensation reaction. So you're removing water, and that's usually coupled with ATP. So that's to cost you some energy. Um, yeah, open up some ATP to do that. What else do we have here? There's also a common naming system, which is a little bit easier to use, but has a little bit of ambiguity. So sometimes it's not 100% clear what you're doing there. So ATP uh, catalyzed by hexose phosphotransferase. That catalyzes the reaction between ATP and hexoses, and the common name for that is just hexokinase. Hexokinase is going to tell you, okay, I'm going to react something here, but really it's a hexose phosphotransferase, so it's the common name is a little bit shorter, but doesn't tell you exactly what it's doing. This one kind of does tell you. I'm transferring a phosphate group um, involving a hexose.